Hello ladies and gentlemen, this is the skin model, and particularly the front side. What I'd like you to do with this is recognize it's divided into three parts. These parts are labeled on the front. So we have the palm or sole, the axillary region or armpit, and then we have the scalp. So you can see these three different regions. Many structures will hold true for all three areas. However, there are differences in all three, which is why we see them separated out. Overall, we take this model and divide it into three separate areas based off profile from superficial to deep. Superficially, from this pink line here up, we have the epidermis. From below that pink line to the top of the yellow stuff here, this middle region is called the dermis. And this area with the light yellow here, or it's thicker over here, is the hypodermis or subcutaneous layer. We're going to focus in on the epidermis. We have A, B, C, D, and E. E's right here on the pink layer. These represent the 5 different layers of the epidermis. A is the stratum corneum, which will be present in all three regions of the model. B represents the stratum lucidium and is only present in the palm or sole of the foot. C is the stratum granulosum. D is the stratum spinosum. E is the stratum basale. E is connecting with the basement membrane to the connective tissue we have down below that. So as we look around, we see A, C, D, and E in the armpit region, and the same as we move over to the scalp region as well. All right. Please note that not all of those structures are in your lab packet unless otherwise instructed. Please know the listed structures in the lab packet. As we move down to the dermis, we can see blood vessels, we can see some tube-like structures in white, and we can see some yellow structures. Now the dermis is divided into different layers that you don't find in the lab packet, but one of those layers has these dermal papillae, which are these finger-like projections that we see sticking up, including this one back here. So here, and they're found in all the layers. These are what represent or create our fingerprints. As we move down, we can see a couple of different dark yellow structures. We've got free nerve endings that come here and just kind of end. We have Mesner's corpuscles that are found up in the dermal papillae specifically. They sometimes look like a pine cone in structure, but tiny. And then down below, we have these round structures, and these are called Paxinian corpuscles. All of these are part of our sensory receptors within the skin. Now while we're on this section here, this is a series of tubes that runs up and comes to the surface of the skin. This is the same set of tubes running up to the surface of the skin that we see here. This is in a cross-section format. This is in a 3D section format. Both of these are sweat glands, and they represent the same type of sweat gland. It is called a merocrine sweat gland or an eccrine sweat gland. A second type of sweat gland we have, instead of emptying at the surface of the skin, empties into a hair follicle. This is the hair in brown with the white center. In pink around that we have the hair follicle. And this is emptying here. Note, we see that we have a tube-like structure here, similar to what we had with our eccrine sweat gland, and that again is our apocrine sweat gland with nerve innervation. And here's the nerve innervation for our eccrine sweat gland. Over here, attaching to the hair follicle, so here and then these things here, we have another type of gland. This is called a sebaceous gland. It will release oil into the hair follicle. It does not go to the surface of the skin. In the middle right here, we have another eccrine sweat gland starting down low, working its way up to the top. So we have apocrine and sebaceous glands that both dump into the hair follicle. The hair shaft is that hair in the center, and it extends up beyond the edge of the skin, like we're seeing in a few places along the top. So J on this model represents that hair shaft, whereas C on that picture right here represents the hair follicle. When our hair stands straight up, it does so because of a muscle that has nerve innervation. All of these tiny muscles connecting to each and every hair is called an erector pili muscle. Now that is the basic structures listed for the dermis in the lab packet. As we go down, we find this yellow stuff, which is adipose tissue or fat. That is its own type of connective tissue. It has several blood vessels running throughout it. The blood vessels would also have lymphatic supply with them. And we see that these blood vessels branch off and they go all throughout the dermis. However, these blood vessels do not go into the epidermis themselves. So if we take a close-up here, what we see is that our stratum basale actually gets good supply of nutrients and oxygen and its living layer because of its proximity to the connective tissue in the dermal layer. However, as we go further out, D, C, B, and A, we lose all that wonderful nutrients and oxygen, so those are dead and dying tissues. So A would be quite dead, D and C are in the process of dying and changing shape, and that covers the front side of our skin model. I do want to show you the back side briefly because it's pretty darn cool. So in this side, we can see what a first degree versus a second degree and a third degree burn look like, how deep the damage is gone. And you can certainly tell the epidermis is gone in a third degree burn. The dermis is highly damaged, and you may have damage all the way down to the hypodermis. Knowing that the stratum basale is part of the epidermis and is what creates tissue layers, it helps us understand why third degree burns are going to require intervention, and even why some second degree burns can require intervention. All right, that's it for the skin model. Thank you for your attention.